Hey BioTeam. So if our cells want to build a protein, the first thing that they're going to need is a gene of DNA. Now, DNA serves as the instructions for building the proteins, but it's not involved in the actual building process. And that's because DNA's double-stranded nature renders it a relatively inflexible molecule. So it's good as a stable source of storing information, but it's bad for actually being able to build things. Plus, in eukaryotic cells, DNA is stuck in the nucleus. It's contained in the nucleus away from where protein synthesis occurs. So what DNA will do is it will go through a process called transcription to build RNA. And since RNA is single-stranded, it's flexible enough to fold in the shapes necessary to actually build the proteins. So in order for RNA to build a protein, what it'll do is it'll travel to the ribosome. And so this faded mass in the background is the ribosome. And at the ribosome, the RNA will perform a process called translation, in which it builds the protein by adding on a single amino acid at a time to form a long chain of amino acids that can then fold into the protein. So biologists will summarize this entire process as the central dogma, or the central idea of molecular biology, this process of how proteins get built. And the general summary is that in order for a protein to get built, you need DNA, but that DNA first has to transcribe into RNA, which is flexible enough to do the translation necessary to build a protein. And that's it. At this point, you guys have some practice problems. We'll see you next class.